Welcome back to the BioFun channel. Imagine a world where we can treat diseases before they even appear. Imagine growing crops that resist pests and drought. This is the promise of DNA technology. It's a powerful tool that allows us to understand and manipulate the very essence of life. This revolution is genetic engineering. It's the process of directly altering an organism's genes. Scientists can add, remove, or change specific genes. This technology is transforming medicine, agriculture, and many other fields. With DNA technology, we are entering a new era of scientific exploration. It's a journey into the microscopic world inside our cells. Here, we can unravel the secrets of life and shape the future of our planet. The possibilities are as limitless as the universe itself. To understand genetic engineering, we need to start with DNA. This molecule is the blueprint of life. It resides within the nucleus of every cell. DNA holds the instructions for building and maintaining an organism. Think of DNA as a vast library filled with genetic information. This information determines our traits. These traits range from eye color to our susceptibility to certain diseases. DNA is composed of 4 chemical building blocks. Scientists represent these blocks with the letters A, T, C, and G. The sequence of these letters forms the genetic code. This code is like a language that dictates the production of proteins. These proteins are the workhorses of our cells. They carry out essential functions in our body. By understanding the language of DNA, we can begin to decode the secrets of life. The first step in genetic engineering is to obtain the desired DNA. This DNA comes from a donor organism. Scientists carefully select this organism for its specific traits. These traits could include disease resistance or enhanced nutritional value. The process begins by breaking open the cells of the donor organism. This releases the DNA. Scientists use a series of chemical treatments to isolate the DNA. They separate it from other cellular components. This purified DNA is precious. It serves as the raw material for genetic engineering. Once we have the DNA, we need a way to cut and paste specific genes. That's where restriction enzymes come in. These enzymes are like molecular scissors. They cut DNA at specific sequences. Imagine wanting to remove a chapter from a book. Restriction enzymes are like finding the exact sentences that mark the beginning and end of that chapter. There are many types of restriction enzymes. Each type recognizes a unique DNA sequence. This allows scientists to cut DNA precisely at desired locations. This precision is crucial for genetic engineering. 2 types of restriction enzymes are used. 1. Sticky end restriction enzymes, which do not cut straight across both DNA strands. Sticky ends can bind to other DNA fragments that have complementary sticky ends. 2. Blunt end restriction enzymes, which cut straight across both DNA strands, produce blunt ends. Blunt ends can join to any other DNA fragment with blunt ends. Now quiz time. Are you still with me? Here's a question for you. What do you think is the wisdom behind God creating cells with 2 different types of restriction enzymes? And how do geneticists take advantage of this diversity? Now please write a comment down below. After cutting DNA with restriction enzymes, we need to sort the fragments by size. This is where gel electrophoresis comes in. It's a technique that separates DNA fragments based on their size. With an electric current, imagine a racetrack for DNA. In gel electrophoresis, scientists load DNA fragments into a gel. At the negative pole, this gel acts like a sieve. When an electric current is applied, the DNA fragments move through the gel. Smaller fragments travel faster and farther than larger ones. This process creates a pattern of bands. Each band represents DNA fragments of a specific size. Scientists use this pattern to analyze the DNA. They can identify specific genes or mutations. Now quiz time. Did the DNA fragments migrate towards the positive or the negative end of the gel? Deduce the chemical nature of DNA. Hint: think about whether DNA is positively or negatively charged. The future of life's code. We've just scratched the surface of DNA technology. The potential of genetic engineering is limitless. As we go deeper into the intricacies of DNA, we unlock new possibilities. From treating diseases to engineering sustainable solutions, the future holds incredible promise. The ability to manipulate the blueprint of life comes with great responsibility. It is our duty to wield this power with care and ensure that it benefits all humankind. As we venture further into this uncharted territory, we must proceed with caution and wisdom. The future of life's code is in our hands. Time for you to act as scientist. Research how gel electrophoresis is used in forensic science for DNA fingerprinting to solve crimes. Highlight its importance in medical diagnostics, such as identifying genetic disorders. If you're lucky enough to be here on live with me now, it's time to get active. Grab your gear and let's dive into the world of genomic scientists. We'll use simple tools to explore how they do their groundbreaking work. Let's get started. Keep curious and keep exploring the natural world around you. Thank you for subscribing to BioFun channel, where your curiosity fuels our exploration of the fascinating realms of biology.